Okay, we're going to start off with Unit 5, which is all about viruses. So the first is to know all of the viral components. So these two pictures are both viruses. The difference between them is the one on the left is enveloped, which you can see right here in this outer area. And the one on the right is known as and referred to as naked because it is actually missing the envelope. If you have your outline next to you while I go through these components, the best way to study this is draw your own virus and then label the components and know what they actually are. So let's start with the inside. Both of them have what's known as genetic material. Later you'll understand why I'm not specifically saying DNA or RNA, because viruses can be both and they have unique genetic material. So again, the inside is just the genetic material. So next, you see these little balls, or little round circles. These are known as the capsid. And the capsid is actually a protein coat, which surrounds all of the genetic material inside of both of them. This is made up of protein, and each individual ball is known as a capsomere. So the one on the left has 1, 2, 3, 4 capsomeres, but if this picture were accurate, these actually go all the way around the entire virus on both sides. It just isn't drawn into this picture. So a capsid, again, is the protein coat that surrounds the genetic material, and each individual little ball of the capsid is known as a capsomere. While you take a look at these two virus pictures, I'm going to make a statement, and hopefully you can understand why I'm saying this. When an envelope is when a virus is enveloped, the envelope is the outermost layer of the virus, as you can see right here. And it is surrounding the capsid made up of capsomeres. When a virus is naked, the capsid are actually the outermost layer because there is no virus. So please make sure that that makes some kind of sense when you are studying this material. So next, you see all of these little spikes. Notice that you have pink ones over here on the left and you have the yellow ones on the right. These capsids are actually known as, these things are actually known as spikes. They are glycoproteins. Glyco implies sugar, so they are made up of a combination of sugars and proteins. And these guys actually project out from either the envelope in an enveloped virus or the capsid in the naked virus because that is the outermost layer. So what these do is these actually attach to receptors on the host cell to allow for attachment to occur, which you'll soon see is the first step of the virus life cycle. I'm going to give you a couple terms that we're going to mention later. So these spikes are hemagglutinin or neuraminidase. When you hear that a virus is H1N1, which you've probably heard when we speak of influenza, the H is for hemagglutinin, the N is for the neuraminidase. So all of these spikes that surround both enveloped and non-enveloped or naked viruses actually refer to the ratio. So if something is H1 and N1, there's just as many H as N spikes. If it is H5N2, there are 5 times more H spikes than there are neuraminidase, so it's a 5-to-2 ratio. This is how viruses can also be classified. The last component that I want to mention, which isn't in the picture, so I'm going to draw this in black so it stands out. I'll draw this like a little boomerang. Both of them actually have it inside of the virus itself. These are the enzymes, and these are what's known as proteolytic enzymes. And this is found in the core, right by the genetic material. And what these function to do is to break down the actual capsid, because again, that's the protein coat protecting the genetic material of the virus to allow it to release and infect neighboring cells. Please make sure that you are able to redraw this picture for yourself, label the different components, and know what they actually do. So if you take a look at this picture right here, there are many different kinds of viruses. Some of them have odd shapes. Bacteriophages we've talked about before during transduction. Notice there are ones that are helical, ones that are hexagons and octagons and icosahedrals, different shapes. You don't actually need to know the shapes, but I do want you to look at the pictures on the right. So here is a naked one. Notice that the capsid, made up of capsomeres, is the outermost layer because naked has no envelope. And then on the right side, this is the enveloped one. And again, this purple right here is the envelope, and notice that the spikes are attached to the envelope. Then internal to that right here, you can see the capsid, because in an enveloped virus, yet again, the outermost layer is the envelope itself. So a term that I would like you to be familiar with is virion. A virion is a complete virus particle. That means it contains all of the components of a virus. I need you to remember this because we're going to talk about something later called viroids, which is completely different and not the same. So please remember that a virion, by definition, I believe it's number 2 or 3 on your outline, is a complete virus assembled particle. Okay, next we're going to talk about age range, or size range, I should say. Viruses are measured in nanometers. They are much smaller than bacteria because bacteria are actually measured in micrometers. This picture right here, this is the host cell. It's telling you it's the human liver cell. Inside of this green guy right here is actually E. coli, so a bacteria. And if you look within an E. coli, 1, 2, 3, 4, 5, 6, 7, 8, 9 different viruses can fit. So this is to give you a size visual to understand that viruses are incredibly small and measured in nanometers. So we're going to go through the steps of viral replication, and then I'm going to give you a little bit of information about each step, and then we'll look at one picture where it puts all of the steps of viral replication together. If you look at the original Unit 5 PowerPoint 1, a few slides from now, there is actually a show where you click the space bar and it'll show this happening in action. You can also Google that if you'd like. This is done on a PDF so that I can annotate the PowerPoint for you, so I'm just going to show you the pictures. So step number 1 in viral replication is known as attachment. Another name for attachment is adsorption, which you can see on the top, and it's also listed as the first step of viral replication on the first page of your outline. So what you're looking at here is this is the host cell. We don't know exactly what the host cell is at this point. We don't know what type of virus this is. This is an enveloped virus, and you know that because this gray area is an envelope, and these green things are the spikes. Notice the spike of the envelope, this is the step of attachment, attaches to a receptor found on the host cell. The pluses and minuses are because there are charges. Not every single spike will attach to every single receptor. Remember, viruses attach relative to whatever their target cell is. So if this were, let's say, an RNA respiratory virus, then this host cell would potentially be lung tissue, but that may not attach to, say, digestive tissue cells. So attachment step 1, you have the spike of the virus attaching to a receptor on the host cell. Couple things to keep in mind. There are 2 different ways that cells can, or viruses can or cannot get into cells. There are 2 different processes, and this is when we start talking about entry or penetration. One of these 2 pictures represents, here, let me switch colors, make that a little simpler. Okay, one of these 2 are fusion, and one of these 2 pictures are endocytosis. Fusion is when you have a fusing of the membranes. So right away you should realize that a naked virus can't do fusion because fusion implies that there's a membrane in both not only the host cell but also the virus. So here you have a virus that is obviously enveloped. You can see the spikes there. This is the membrane of the host cell. Here's the receptors that those spikes will actually attach to. And if you take a look at the color, the membrane or envelope of the virus is purple. The envelope or plasma membrane of the host is orange. If you look a little further down this picture, the 2 are fusing together because this orange right here represents the host membrane, whereas the purple represents the envelope of the virus. So right here, the two have fused together, making this top picture fusion. Then the virus is actually going to enter. Here, the proteolytic enzymes are at work to release the genetic material into the host cell. If you take a look at the bottom picture, here there is no meshing together or fusion of the membranes. You have an enveloped virus. It is invaginating. This orange is, again, the host plasma membrane. And now this is very hard for your immune system to detect, because once it's done invagination, which is also endocytosis, you have a virion, or a full virus particle, and notice that it is enveloped because it fused in, or I should say meshed in, invaginated, doing endocytosis, and now the outermost layer, the orange, is related to the host plasma membrane. At some point, again, the proteolytic enzymes are going to work to help release the genetic material into the host. So please remember, fusion implies that the two membranes fuse themselves together, like you can see in this picture, and endocytosis is an invagination where nothing fuses together, but in fact the virus particle invaginates and actually picks up the membrane of the host. I'm going to skip this one here. So I keep saying genetic material, and I said that I would later explain why I am using the term genetic material and not talking about this one's a DNA, this one's an RNA. There's a reason for that. Viruses are very unique in their genetic material. So traditionally DNA we know as double-stranded, and RNA we know as single-stranded. But in viruses, DNA could be single-stranded, double-stranded, or even circular. Whereas in RNA, what we normally know as single-stranded, it can also be double-stranded and segmented, which is broken up into little pieces. So it's incredibly important that you remember, on a virus, unless if you are told, it must be referred to as genetic material. You can't say it's DNA or RNA, because unlike with other organisms, if it's double-stranded, you can assume DNA, single-stranded, you can assume RNA, but with viruses, that is not the case. They're very unique. So unless it says this is an RNA virus or this is a DNA virus, by visually looking at a picture, you have to refer to it as genetic material, because you just simply don't know, because there are so many different variations. So this is the picture that you can click after each step. If you look on the original PowerPoint, I can't do that for you, so I'm just going to go through the steps. So again, here we have the first step, which is attachment. And attachment is where the spikes of the virus attach to receptors on the host cell. Another term for attachment is absorption. Then our second step is penetration, which is also known as entry. This is when the virus is entering into the host cell. Then we have a third step, which is known as uncoating. This is where those proteolytic enzymes work. And what they do is they break down the capsid like this to allow it to release the genetic material. On your exam, you're going to see a picture similar to this for viral replication. The picture is going to show up 6 to 10 times, and it's going to ask you what's happening in all of the different steps. So after the third step of uncoating, we have number 3 and 4 going on. And number 3 and 4 are synthesis and maturation. So number 3 is up top here. Synthesis is actually all of the different steps. Now remember, viruses are intracellular. So again, intracellular obligate parasite, which means they cannot survive without the host. So the host functions attached to number 3, which is synthesis. Synthesis is replication, transcription, and translation. The virus takes on the host functions and whatever it happens to be doing. Then the next step at this point comes down here, which is assembly and maturation. I see this as the virus making its own little army. It starts replicating and making virions, which are, again, complete viral particles. We talked about that right in the beginning, where they're fully assembled. And then the last step down here, we'll call it number 5, is release. Release is when the virions, or complete virus particles, have already assembled and matured, and they're released to infect neighboring cells or host cells. If you take a look at this picture right here, the one on the left, which is this guy, and the one on the right, which is this guy, they are stolen from two completely separate books. So if we were in class, I would ask you to compare these two pictures, as you can see right here, and ask you which one is actually better. And the answer is neither's better because they show the same thing. Most students tend to like this one on the left only because it's a little bit more clear, a little bit more writing, and I guess they're more comfortable with the picture because it's simpler and more colorful. But I'm going to use the one on the left because it's larger to explain to you what's actually happening. So, if you take a look right here, you have an enveloped virus. I know it's enveloped because of that purple covering right around the virus. And then I want you to take a look at the spikes. I would ask you in class, is this a DNA or an RNA virus? Some of you will quickly say, well, I don't know, you have to tell us. Look carefully because right here it says it is a DNA enveloped virus. So again, you have the first step of attachment, where it is attaching to the host cell. Then you have entry occurring, or at B, we can write between A and B, is penetration. Between these two steps is when you actually have the uncoating being done by the proteolytic enzymes. So again, those enzymes are breaking down this capsid to release the genetic material, which we now happen to know is DNA because it's written right there for you. Next, we have synthesis occurring, which is replication, transcription, and translation. So translation's making the proteins. Replication, you see that the DNA is in fact being replicated. And for transcription, you should see that mRNA is being made right there. So after the steps of synthesis, you end up with lots and lots of viral particles being made. So this is now the next step of maturation, or you can also call it assembly if you remember. And then the final step is right here, which is release. So this is exactly the same as the previous picture. You could study either of them. So now let's take a picture here. Let's take a look at the picture on the right. Okay, so you have a virus. Is it DNA or RNA? So if you're taking a look, you see right here it says DNA. It's referred to as a virion, which is a complete virus particle. You have absorption or attachment. This one is telling you it's fusion. So what's fusing? The envelope of the virus with the plasma membrane of the host cell. Next you have penetration, which is also known as entry. And then you have uncoating, which is done by the proteolytic enzyme. This is breaking down the capsid to release the genetic material. Next we have synthesis occurring, which is everything from replication, transcription, and translation. Please remember these steps of synthesis are occurring for the viral genes. Next we have assembly and maturation, where the army is being made of virions, which are complete viral particles. And then finally the last step of release. So hopefully you can see that both of the picture on the left as well as the right are showing you the exact same information. So a couple terms that I need you to remember are the proteolytic enzyme is working to help release the genetic material from the protein coat of the capsid. I need you to consider if it is fusion or endocytosis. If I were to ask you if endocytosis or fusion can occur with a naked envelope or a naked virus, the answer is absolutely not, because fusion is again the envelope of the virus fusing with the plasma membrane of the host. So if it's a naked virus, there is no envelope. So naked viruses can only invaginate and do endocytosis. And lastly, it's important that you remember that the term virion implies and refers to a completely assembled virus particle. So if I were to simply tell you these key steps again, I would say that virus has the steps: attachment, penetration, uncoating, synthesis, and assembly and maturation, and finally it's going to release. When the attachment and penetration is happening, the spikes of the virus are attaching to receptors on the host because the goal is to inject its genetic material into the host. When uncoating is occurring, it requires a proteolytic enzyme to break down the protective coat of the capsid to actually release the genetic material or the viral genome into the host. Then you have synthesis occurring, which is replication, transcription, and translation. After those steps, you have assembly and maturation, and finally, the newly created, fully assembled virus particles known as virions are going to be released to then affect the neighboring cells.